Hello viewers, I am Dr. Chaudhary SG from Department of Microbiology, Venkatesh Mahajan Senior College, Usmanabad. Welcome again to my channel Learn Microbiology. In this video, I am going to talk about certain points regarding the enzymes. I have already talked about the types of enzymes, but I have included this slide about that topic in this video. The point to be covered during this video are types of enzymes, properties of enzymes, and specificity of the enzymes. The types of enzymes: endoenzymes, exoenzymes, constitutive enzymes. I am not briefly talking about this in this video, but let's have an overlook. Endoenzymes that are produced within the cell and having activity inside the cell. Exoenzymes that are produced again within the cell but having activity outside the cell. Constitutive enzymes that are produced naturally by the cell irrespective of the substrate concentration or presence of substrates. Induced enzymes that are induced only in the presence of substrate. So now let us briefly continue with the properties of enzymes. Certain properties, that is the colloidal nature, catalytic nature of the effectiveness of the enzyme, reversibility of the enzymes, heat sensitivity and sensitivity to the pH of the enzymes, then specificity of the enzymes. Discuss one by one. The colloidal nature. Enzymes totally or partially made up of protein molecules. We already know this. They are large molecular weight compounds and having very low diffusible rate or diffusion rate, and that's why they forms colloidal system in aqueous media or in water. Being colloidal nature, the enzymes are non-dilysable molecule, but the non-protein part or coenzymes that can be dilysable or dissociable from the apoenzyme portion. They are a very large molecule as compared to the substrate or the group on which they are acts. So these are the properties regarding the colloidal nature of the enzymes. Then effectiveness of the enzyme or catalytic nature of the enzyme. What is effectiveness? It is the ability of enzymes to convert the substrate into the product, and it is nothing but the catalytic power of that enzymes. Enzymes are effectively catalyze conversion of the substrate into the product irrespective of its concentration. Means very low concentration of enzymes can convert substrate into the product. It is the catalytic nature, and this catalytic power or effectiveness is measured in the terms of turnover number. The turnover number is a number of substrate molecule converted into the product per unit time by the enzyme. Okay, so the catalytic power or this effectiveness indicates the activity of that enzyme, and it is measured in the terms of unit. So 1 unit enzyme activity is nothing but the amount of enzyme which catalyzes the transformation of 1 mole of substrate into the product per unit time, and it is conversion is carried out under defined conditions or under the conditions of that media. So next property of the enzyme is reversibility of the enzymes. We know enzymes catalyze the reaction, they speed up the reaction, they catalyze both forward and backward reactions. So this ability means enzyme can catalyze forward reaction or backward reaction is nothing but its reversibility of the, or it is nothing but the reversibility of that enzyme. For example, enzyme lipase can carry out the synthesis of fat or lipid using glycerol or fatty acid, or on the other hand, the enzyme lipase able to hydrolyze this fat into glycerol and fatty acid. So both synthesis and breakdown of lipid can be carried out by the enzyme lipase. So this is nothing but the reversible reaction. Okay. It is not possible in case of all enzymes. The exception to this is urease enzyme. What is the function of urease enzyme? Urease hydrolyzes urea into ammonia and carbon dioxide, but it is not involved in the synthesis of urea. The synthesis of urea is carried out by another type of enzyme, that is arginase. Arginase enzyme synthesize urea using arginine as substrate. The next important characteristic is heat sensitivity. Being protein in nature, enzymes are highly sensitive to the temperature. At higher temperature, the protein nature of the enzyme get denatured and the enzyme become non-functional. Their properties are not restored even when coming back to the normal temperature. But at very low temperature, enzyme temporarily inactivated, but it does not get denatured. After coming back to normal, the activity regain back. The rate of enzyme activity increases with rise in the temperature. There is a certain temperature for each enzyme at where the enzyme activity is maximum, and this temperature is nothing but the optimum temperature of that enzyme. So every enzyme having its optimum temperature. As like temperature, the pH sensitivity, enzymes are sensitive to the hydrogen ion concentration, and this hydrogen ion concentration is nothing but pH of that enzyme. So every enzyme acts optimally at certain pH at where the activity of that enzyme is maximum. So at where the activity is the maximum, the pH is also termed as optimum pH. Below or above, at the both side of the optimum pH, the enzyme activity become lowest or lower. So enzymes having a particular optimum pH, depending upon the optimum range, the enzymes can be active at alkaline pH or at acidic pH or at neutral pH. So it varies according to the enzymes. Certain enzymes are active at alkaline pH. One example I quoted here, phosphorylase enzymes that activate at alkaline pH, while as glucose phosphate enzymes involved in the conversion of glucose phosphate to starch are active as acidic pH. Enzyme specificity. So enzymes are having specific nature for their catalysis. Enzymes are highly specific for their reaction, for their substrate or the group they acting upon. Reaction, substrate or the group acting upon. They can specific for the forms or they can specific for their site of action. So we will talk briefly about the specificity of the enzyme in next video. So This is about the properties of enzyme.